Welcome to the BioWhisperer channel. Our topic today is on a perspective overview of the select agents associated with pathogenic diseases. So let's discuss in this short video an introductory perspective on the key concerns associated with infection transmission linked to select agents. Select agent refers to biological agents and toxins that have been determined to have the potential to pose a severe threat to public health and safety. While potentially dangerous, work with select agents and toxins provides important scientific discoveries that have led to improved detection, prevention, diagnostic, and treatment options for diseases considered to be some of the most threatening to public, animal, and plant health. It also covers animal and plant health, including animal or plant products, and examples include the toxin ricin, the virus Ebola, the bacteria Yersinia pestis, and anthracis, amongst others. SARS, or severe acute respiratory syndrome, was first reported in Asia in 2003, nearly 2 decades back. The coronavirus that causes it, called SARS-CoV-1, has spread to more than 24 countries across 4 continents before the global outbreak was contained. MERS, or Middle East Respiratory Syndrome, is viral respiratory illness that is new to humans. Contact with camels is postulated to be the cause, where it is first reported in Saudi Arabia in 2012 and spread globally. MERS-CoV infection is associated with severe respiratory illness, including fever, cough, and shortness of breath. Here are some of the images from the Google search screen grab on MERS. COVID-19 affects different people in different ways. Most infected people will develop mild to moderate illness and recover without hospitalization. First reported in China in 2019 and spread globally, with recurring variants such as Delta and Omicron as variants of concern classified by the World Health Organization. In response to infection control, countries have adopted vaccination, therapeutical and social distancing strategies are developed and deployed at unprecedented phase, including fast-tracked review and approvals of vaccines and therapeutics coordinated by the World Health Organization. Exposure and incubation of a causative pathogen typically follows the infection timeline. The time between initial exposure to the virus and the appearance of symptoms is known as the incubation period. This period is typically around 4 to 5 days, although it can last up to 2 weeks, or perhaps even longer in rare cases. During the symptomatic phase, the subject is infectious and can transmit and infect others. Recovery proceeds and may take up to weeks. I would thank you for helping to share your love for this science channel by clicking the like button and subscribe for future updates. With that out of the way, let's continue the discussion. As for the considerations of the select agent, scientists consider 3 aspects. Firstly, the effect on human health of exposure to the agent or toxin, followed by the degree of contagiousness of the agent or toxin and the methods by which the agent or toxin is transferred to humans. Third, we consider the availability and effectiveness of pharmacotherapies and immunizations to treat and prevent any illness resulting from infection by the agent or toxin. Within the US CDC, the National Select Agent Registry Program declared SARS coronavirus a select agent in 2014. We define a select agent as being a bacterium, virus, or toxin that has the potential to pose a severe threat to public health and safety. Hence, studies or research performed on select agent needs consider the natural risk of different infectious or potentially infectious diseases or toxins, the type of research being conducted on these diseases and toxins, and the amount of the agent or toxin being used in research. These safety and security requirements are in place to protect the health and safety of research staff and to reduce or eliminate exposure of laboratory workers and the outside environment to potentially dangerous diseases or toxins. In terms of working with select agents, it will require higher scrutiny, including added responsibilities and due diligence in keeping records. Proper authorization is needed as well, since select agents and toxins can pose a severe threat to public, animal or plant health, or to animal or plant products. Having the records kept for 3 years as well as having relevant inventory management and training will be essential when working with select agents. Now, let's take time to ponder over one of the notable example. Smallpox, a deadly virus infection, causes fever, convulsions and painful blisters all over the body at a fatality rate of 30%. Intensive vaccination campaigns wiped it out in Africa, Asia and South America. The virus can be transmitted by direct contact with the fluids within the scabs as well as by contact with body fluids of a person with smallpox or items they have touched. Soon after smallpox was eradicated, the World Health Organization consolidated the remaining stocks of variola virus to 2 labs, the Russian State Center for Research on Virology and Biotechnology and a lab at the Centers for Disease Control and Prevention in Atlanta. The intent is to keep for development of countermeasures and to develop therapeutics, a good idea that yielded a therapeutic product. These countermeasures that derived from continued and ongoing research could potentially be useful as stockpiles in case of bioterrorism attack. Smallpox, tecovirimat, the first drug with an indication for treatment of smallpox. Though the World Health Organization declared smallpox, a contagious and sometimes fatal infectious disease, eradicated in 1980, there have been long-standing concerns that smallpox could be used as a bioweapon. Let's ask ourselves the open and difficult question. The risks is no doubt present, expected or unexpected. Just like the explosion at the lab facility holding and storing live variola virus stocks. Mitigating measures, greater accountability and definitely a global debate on the way forward in a consolidated collaborative manner will be important. Let's rethink about the smallpox issue and the unexpected lab explosion. It is unavoidable that members of the global community would be worried about potential release and outbreak. Rethinking it in the perspective of other infectious diseases such as mumps, polio or even COVID-19 causative agents after the pandemic. What should we do with the causative agents and how will health agencies and inter-agency coordination look like in handling, storing of these strains? My guess is the World Health Organization will be in the lead to organize the consolidation and removal of stockpiles. In fact, like any past infectious diseases, open sharing of genomic sequences in databases permit global collaboration and readily availability of information in databases to researchers. In fact, full annotated genomic sequence information are publicly available. What happens if Rouge scientists decided to do otherwise with these rich resources? We have a video on dark biotechnology which you might also wish to review. Given the rapid rise of synthetic biology, a futuristic field of science that seeks to master the machinery of life, has raised the risk of a new generation of bioweapons which might mean advances in the area. Mean that scientists now have the capability to recreate dangerous viruses from scratch, make harmful pathogens even more deadly. The threat of bioterrorism meant that authorities have to rethink the current approach of making information public and the potential of Rouge scientists with bioterrorism in mind. For our closing remarks, we brought forward the importance of post-pandemic discussions on what health authorities should do with the research stockpiles and live viruses. Previous case example of smallpox was mentioned. Looking forward, it is about foolproofing any interagency collaboration on what to do with these select agents and to legislate relevant regulations accordingly. Like everyone else, as a global citizen, we are looking forward to the new post-pandemic normal. Will a dream vaccine or therapy be a possibility? We will visit that in our next video. See you then. I would thank you for helping to share your love for this science channel by clicking the like button and subscribe for future updates. If you like this video, please smash the like button and subscribe to this channel for future updates.